So back to lecture 5. Now you can see it, I hope. We're going to cover a little bit of stochastic SIR models and then a few other model structures and then work on a project together. Coming back to comments on project proposals, just emphasizing that for people who hadn't had a chance to develop their project proposals as much as they liked, I'd like to have people hand in revised versions for next week. I'm particularly interested in trying to understand what the possible hypotheses and biological context are, and ideally a couple of different possible papers that you could combine to generate something beyond a single replication. And if you have an idea of what the network structures look like, include that so we can be sure that it's something that's practical to do with tellurium antibody. And also thinking about how to make the project useful as a NanoHub app, educational application. As I was saying, for comments on homework, it's great to use Jupyter Notebooks for your submissions. But if you do that, please put the text into a markup cell. If you go into NanoHub Tellurium, you'll see that if you click on a cell, you can change the cell type from Python to a markup. Within the markup cell, you can type equations in TeX. You can include figures from outside. The exact formatting rules are a little bit complicated because even though the equations are in TeX, the other formatting is not in TeX, which is a little confusing. But I found this nice reference, which is a study, a cheat sheet from IBM that gives the formatting guidance for typing into Tellurium markdown cells. And then again, if you're for your project proposals and elsewhere, if we're talking about papers, I think I said this the other day, please do that beyond just the name of the paper and the link. It's helpful to have the link, but it's also helpful to have the bibliographic reference. And I'll point out that if you go to Google Scholar and you type in, if you open up the reference list that you're looking at in Google Scholar, there's this double quote button in Google Scholar. If you click on that, it will pop up this cite window, and that cite window will give you the citation for the paper you're looking at, and you can then click on that and select it. And so you don't have to retype anything to get the full citation. So I'm not asking people to spend a lot of time retyping citations, but it is helpful to have the full citation. Most of the time, the papers that people have been referencing are ones that are unique names, but occasionally they're not, and it's always helpful then to have that full citation. If you're going to write a paper and do something later on with it, you'll need it anyway, so it's helpful. If you're going to use footnotes and you do things in Google Docs, there's a package called Zotero, which is a free download. That's quite a good citation manager that I recommend. Zotero, unfortunately, is not available for Word. So if you're in Word, you could use something like EndNote otherwise. I don't know if anybody is a tech enthusiast. I'm not a particular tech enthusiast, but if you use Overleaf, then you can export citations from Google Scholar as BibTeX.
Dr. Glazier.
Yep.
Is there a specific citation style we should use?
No, which any of these are fine. I don't have a particular preference. It's just that some people when I ask them for paper lists, very nicely gave me a list of 10 papers with just the titles. And so then I had to do a certain amount of Googling to find the papers. And so how it would have, it's not a big deal, but it saves everybody a little bit of time if you have a full citation from the beginning. You're welcome to use any citation format that you like. I'm not addicted to any one of them or allergic to any one of them. So let's come back to the class topics. The order may have changed slightly from what I said before. I'm going to just quickly remind us what the thing we mainly did last time was repeating that exercise from the time before, using the incidence data and active case data to try to get estimates of the clearance rate mu, the recovery rate mu, and the R naught. And we did that by fitting first the tail of the active cases to try to estimate the recovery rate, and then fitting the leading edge of the curve on a log scale to try to get the incidence rate. Once we had N gamma on the front and mu on the back, we could estimate R naught and also potentially S naught, the effective fraction of the population that was susceptible and exposed, susceptible to being exposed. Some of the papers that people have been looking at talk in a lot of detail about how to use the clinical experimental data that are available. And it gets pretty complicated to try to interpret these data. But at least we had that basic estimate, and the numbers we came up with weren't too bad. People put them in that table, which is nice. We also talked about the concept of herd immunity. And the key idea of herd immunity is that if the basic reproductive rate of the virus is R naught, if the fraction of the population that is susceptible is less than 1 over R naught, then the disease dies out on its own. So if you have a disease like influenza with a typical R naught of 2, then if half the population either has recovered from the disease and has immunity from that or has been vaccinated, then it doesn't mean that you won't have people get it and that it won't spread locally, but you won't get epidemic transmission through the population with exponential increase. And so in general, diseases stop on their own, at least as epidemics, once the fraction of the population goes down below, susceptible population goes down below 1 over R naught. Of course, if R naught is big, like 30, then 97% of the population has to be immune to stop the spread of the disease, which is a problem. And that was a little homework problem to look at that. And we had pulled up some numbers from various places, Wikipedia and elsewhere. And we talked a little bit about strategies, and there was a nice paper that someone sent around talking about some of those strategies, reducing the number of people that you contact when you're infected, reducing the probability of infection per contact by wearing a mask or other distancing, reducing the time over which somebody's infectious. That's one way to do that, is to get better sooner. Another way to do that is to quarantine people when they're sick. And then classically vaccination to reduce the fraction of the population that's susceptible in the first place. And in this particular little table, which was from a paper, online set of slides that I found by somebody named James Holland Jones, he took R naught, got 1 over R naught, calculated what fraction of the population you have to vaccinate to make things work. And again, measles, where R naught's 20, you have to vaccinate essentially everybody. Influenza, you can do a little bit better. of course, we've had a pretty decent influenza vaccine for a long time, and we still have influenza epidemics. Well, not this year, but every other year we've had influenza epidemics. And so the availability of a vaccine doesn't necessarily mean enough people get vaccinated to prevent the spread of the disease. Smallpox is the opposite. In the case of smallpox, vaccination was successful to eliminate the disease completely. People think maybe polio is on its way out, although it's not so obvious. One of the issues, of course, with vaccination is whether there's an animal reservoir for the disease. Because even if you get 75%, 80% of the population vaccinated, if people are exposed to animals that carry the disease, then that's you can reinfect. And so if there's a lot of transmission between a common animal and humans, then it's harder to get rid of the disease. If the virus is confined to a particular species of bat that's very localized to a particular place, then it's easier, like in Ebola. If it's a virus that's exchanged between humans and pigeons or cats or dogs, then it's harder. And then we talked a little bit about parameter optimization. Josh very kindly gave us some lecture on that. We did a little bit of basic parameter optimization. The key idea of a chi-squared or residual, which is we look at each data point in our series, our experimental data points and our simulated data points. We measure the distance between them. We do a sum of the squares of those distances and try to minimize that. There's some art to that. Chi-squared isn't the only distance function that one wants to use. And we'll find out later that under some circumstances, chi-squareds are not the best way to do it. The key concept we had here was that there was some idea of some set of parameters. For each set of parameters, there was some deviation between the simulated and the experimental data set, which we characterized by the subjective function. The higher that value is, the worse our fit is. The lower that value, the better. There's a global minimum here, which is our best for any possible set of parameters. There's a dip here and here, which are local minima. And one problem with many optimizers is that they find local minima and they don't find global minima. Another one is that if you start out very far away from your optimum, changing the values of the parameters doesn't change the objective function very much. So if I'm sitting over here near the arrow which says objective function, this surface is very flat, which means as I change my parameters, my objective function doesn't change very much. So I don't know which way to go to get a better result. And so you'll find that if you don't start fairly close to the place you're trying to go with an optimizer, you'll wind up having the optimizer churn and do nothing or get stuck at a local minimum. That can be pretty frustrating. And so the reason we went through that whole elaborate exercise last time and the time before of hand fitting these initial parameter values was that by guessing initial parameter values that are pretty good, we could get close enough to this optimum that our optimizer can take us down that hill. Whereas if we started way over here, the optimizer doesn't know that our optimum is over here. Optimizers aren't magic. And so this is something that you run into a lot and we'll run into more and more as we go on when we do fits, that there is no magical way to optimize parameters. But I wanted just to redefine global minimum and local minimum in the context of optimization. Another concept that haven't really worked on so far, and these slides are a little bit out of order, but I wanted to at least introduce the terminology, is the concept of a fixed point. And a fixed point is a concept that comes from dynamical systems mathematics. So we're going to be always writing sets of differential equations. Here they're going to look slightly different from the way they're written before, but conceptually they're the same, which is d by dt, the time variation of variable here with the subscript x1 could be I1, it could be n, it could be s, something else, is some function in the state of the system. So x1 is a function, the x1 dt is a function f1 of the state of all of the other variables. A fixed point, which is usually denoted with a star, and here it's denoted as a vector, although we could write them separately, is a set of values of our variables, x1 star, x2 star, x3 star. In our case, for our SIR model, it would be S star, I star, R star, for which all of the time derivatives are 0. That is, if we start out with a value S star, I star, R star, our equations have 0 on the right-hand side. That is, they stay fixed. So there's the values stay constant. So these are steady state values. And we need them all to be 0, all the derivatives to be 0 simultaneously. In the case of our SIR model, the SIR model has slightly strange behavior in terms of fixed points. If I look at my equations, everything in this model is slave to I. So if I set the number of infected individuals to 0, the second term, di by dt, is 0 because both of the terms in that equation are proportional to I. And so if I star is 0, ds by dt is 0 because if I is 0, this term is 0. dr by dt is 0 because mu times I is 0. So any value of s, comma, I star equals 0, comma, any value of r is a fixed point of that SIR model. That's sort of a trivial fixed point. What it means is that if nobody ever gets infected in the first place, there's no infection. That's pretty boring. The second one is a little bit more interesting, which is suppose that I was not 0, to begin with at least. These guys are only going to be non-zero if I is 0. But suppose we could ask at least if I is not 0, can di by dt be 0? And that's how we found the case of herd immunity. Because if we say I is not 0 and we want di by dt to be 0, then we need n gamma over s naught times s minus mu to be 0. So in that case, we have a fixed point where S star is equal to n gamma over S naught times mu. So that gives us I star fixed. The fixed point there isn't very interesting. When we come back to it in the context of SIS models, it's more interesting. But I wanted to just define what a fixed point was so that you'll have heard of that. The next thing I want to do, and it was the one thing I really wanted to do in class before we went on and worked on papers together, was to talk a little bit about stochasticity. So in our SIR model, we wrote an ordinary differential equation, and the number of individuals changed continuously. It wasn't an integer. We could have had 2.5 infected people and 3.1 recovered people. In reality, infection comes in granularity of individuals. The idea that every infected person infects exactly R naught people is also not right. One person who's infected may be isolated and infect nobody, and another person may go around and infect hundreds of people. And so there are fluctuations, and people do agent-based models and chain-based models, Markov models of infection. And what you find is that in general, if the values of mu and R naught are the same, the pattern of infection is similar, but it's not identical. At the end of this set of slides that I keep using, there's that lecture, very good lecture from a group in California, which dug into the analysis of stochasticity a lot and how it changes things. A couple of the papers that people have sent to look at talk about stochasticity. Really dealing with stochastic models in detail is pretty elaborate. Tellurium has one stochastic solver, and it uses an algorithm called the Gillespie algorithm. And the Gillespie algorithm is designed to model chemical reactions. And actually, infection is not that much different from a chemical reaction, because for two atoms to react, or two molecules to react, those molecules have to move around randomly. They have to find each other, so they have to come into contact. And then they have to decide, do I react or don't I react? And for infection to occur, two individuals, one infected and one uninfected, have to find each other. And then we have a probability beta that once they found each other, they get, you have, or gamma, sorry, in this case, they're in fact, you get the infection. And so that's actually rather similar to a kind of Gillespie model. In the classic Gillespie model, you calculate the time interval between events, the time interval between individual chemical reactions. And that means that the output of a Gillespie model doesn't have uniform time intervals the way we do in the simulations we've been running before. Instead, you get reaction time 1, reaction time 2, reaction time 3, and then the changes of the numbers. And it also means that when you do amounts, you have to actually use the real amounts. In other words, you can't use the fraction of the population between 0 and 1. The number really means the number of individuals. So if it's molecules and you have a mole, 6.02 times 10 to the 23rd individuals, you can't simulate. So Gillespie algorithms only work when you have relatively few individuals. So that's something to think about. Gillespie isn't the perfect algorithm for infectious modeling, but it's actually not that bad when you dig into the mathematics of it. And it's easily available to us. So I wanted to do a little bit of Gillespie modeling today before we dig into the other problems we're going to work on. So if you want to run Gillespie in Tellurium, it's exceedingly easy. Instead of typing m.simulate, open parenthesis, start time, comma, end time, comma, number of points, you type m.Gillespie, open parenthesis, start time, comma, end time. And you don't specify how many time points you want to use, because that number of time points is an emergent property of the simulation. So each time you run the same simulation, you'll get a different number of points. There is an option in the Gillespie solver to force it to use a fixed time step. And if you're going to aggregate data sets to say compare them or put them on the same plot, that can be useful. But the bare Gillespie algorithm is that. So what I'd like to do now is have everybody boot up Tellurium. We're going to need it anyway. And I would like them to pull up one of their SIR models that they've played with, anyone you like. And then I want you to replace m.simulate with m.Gillespie, start time, end time, and see what happens. We've typically started with S0 on the order of 10 to the 6th, 10 to the 7th. You don't want to do that, or you'll be sitting here for a long time and you'll get a result that's not very interesting. So I would recommend setting S0, the initial population of susceptibles, to 100, the initial number of infected individuals to 1. And I'd use an R0 of 2 or 3 to begin with. Is that a clear exercise? Everybody? So why don't I give people, sorry?
Where do you want us to change this?
So open up one of our SIR models.
Oh, okay, just any SIR model.
anyone that we've run, which gives you a time series. And then go and take the line, which is m.simulate. Simulate. You don't have to change the m.plot. Plot. The m.simulate, simulate, replace the m.simulate simulate start time, comma, end time, comma, number of points with just the last be start time, end time. You don't specify the number of points. Let's see what happens. And I'll show you code to do it in a second, but I thought by now people are probably used to running SIR functions models, so I'll let you try it on your own for a minute. And we do have a copy of it. If you get frustrated, in the online folder of exercises, there should be exercise 5.1 that you could upload. That'll do it. So why doesn't everybody just take a minute or two to try that?
What would I put in for start time and end time? Because for some reason it's giving me an attribute error at s is equal to m.gillespie. Oh wait, I forgot an I. Never mind.
So instead of having 3 arguments, there are 2 arguments. So it's just start time, whatever your start time. So if you ran your original model from day 0 to day 450, you'd say just keep 0 to 50, but instead of 0, 50, 1,000, just be 0, 50.
Yeah, I realized that I spelled Gillespie wrong. It was just Gillespie without the I in it.
There's actually rather a lot one can do with these stochastic models. I had originally planned some more exercises, but I don't want to lose too much time on it. But I think it's important enough that I want to come back to. I want to at least do a little bit of it now. Especially before we leave epidemiological modeling. There's a tendency on the part of people who do this kind of modeling to assume that the ordinary differential equation models describe reality. The reality is always stochastic. The reality has always got discrete individuals. Now, if you've got a mole of atoms, changing 1 mole 6.02 times 10 to the 23rd plus minus 1 atom probably does, it really is pretty close to continuous. But if you're talking about chemical reactions within inside a cell, and they involve DNA, well, you have 1 copy or 2 copies of your DNA. Therefore, assuming that n equals 2 is continuous infinity is not really a very good approximation. Many chemical species inside individual cells exist in copies of numbers of 2, 4, 10, 15. And so to assume that you can then approximate those as continuous chemical reactions is not right. And so one of the interesting things about biological circuitry, which we're not going to cover so much in this class, is how do these circuits work in the presence of a lot of stochasticity. People who do neuroscience have this problem in spades because your neurons fire action potential. Those action potentials are stochastically timed. How do you do accurate computation with exceedingly noisy, spiky signals? Things like echolocation of a bat. How you do that with random spike trains is almost impossible to think about. You have to have temporal accuracies of microseconds when the spikes have intervals between them of milliseconds, and the spike durations and the jitters are milliseconds. It's actually, some of those are relatively unsolved problems. Okay. So was everybody able to get something like that to work? Okay, so here's what my little model looked like. I put a little title on the plot, although I should probably have actually truncated the strings so that they look better. And I ran 10 replicas. So I said for n in range 0, 11, 1, so I'm going to run it 10 times. Reset it, run the Gillespie, plot it, and overwrite the plot. And what you see is that for the values that I gave you here, I get different results. In this case here, up at the top, there was one replica of the simulation. And so replica, if you run the same exact parameters in initial conditions, each time you run the simulation, that simulation is called a replica. And so here I've run 10 replicas of the same simulation. And if I ran the same ODEs 10 times with the same parameters, I would get exactly the same thing every time. So I would just see one line. Here I see one case where the number of susceptible individuals went down a little bit and then stabilized. And that's because the number of infected individuals stayed low, went to 0 and stopped. In other cases, most of the people got infected, but in some of the runs, 80% of the population got infected. In some of the runs, 95% of the population got infected. And in this blue one here that's hiding in the back, 100% of the population got infected. So I could then have done a histogram, for example, of what fraction of the population got infected. But what I'd see here is that starting with 100 individuals who could get sick and 5 people who were sick, I have a huge range of possibilities for basic reproductive ratio of 2.5, which would be comparable to flu. In one subgroup of 100 people, 5 people got sick, or 10 people got sick, and everybody else was fine. In one group, all 100 people got sick. And in most groups, somewhere around 90 people, 80 to 90% of people got sick. So that shows you that this is quite different from the prediction that I would get from doing the OD. Why don't you try going back to, for the same parameters, go back and plot on top of this. You could do one run where you did an S equals M dot simulate, plot it, and plot the deterministic series on top of it and see where that lands. In this case, what you'll get is something that actually will come right in the middle of this cluster of possibilities. But it shows you that even for 100 individuals and 5 initially infected individuals, the fluctuations are quite large. Did everybody get something that looked like that? Did anybody say it's something that looked totally different? I can do that myself. Let's actually run. Let's do it here. Let me plot the deterministic one. Oops, I didn't. So, and I should have done that. So I can't really see my deterministic one very clearly in the middle, can should be hiding somewhere. Looks like it's hiding right here towards the bottom. So let me plot them. Let me plot them separately. Be easy to see. Okay. So the answer is that the typical simulation among my stochastic simulations looks pretty similar to my ODEs, but there's some wild outliers. There's some cases where much less of the population gets infected and the infection dies out, and there's some where things happen faster. Did people get something that looked comparable to that? Okay.
Yes.
Okay. So now you could try changing S naught and changing I naught and seeing what happens. If you make S naught bigger, does the stochastic fluctuation get bigger or smaller? And in principle, we could do all sorts of things with parameter scans, but we can do this by hand. So here I started out with 100 individuals. Let's make it, everybody do this together, make it 1,000. Notice as I go to 1,000, things get to be smoother. I didn't have any cases where my infection died out completely, but there's still quite a range of the timings. Some of these things happen fast, some of them happen slow, and there's a distribution of the timings around my initial my ODE simulation, and the final number of infecteds is somewhat variable too. So if I go to here 1,000 population, the stochastic simulation is now getting closer to the deterministic one. If I go to 10 to the 4th, at some point this will get so slow that it's not very helpful anymore. Actually, I went to 10 to the 4th and it didn't change much. In fact, it looks like the stochasticity went up a little bit. Notice that there's one case here, this orange line on the right, everything seemed to happen a lot later. So going to 10 to the 4th individuals doesn't necessarily mean you're not stochastic. You could ignore the stochasticity. Let's see what happens if I increase my number of I naughts. Start with 50 infected individuals. Now my variation is much smaller. There's still some, but I have almost a uniform number of people infected at the end. What if I went the other way? And what if I started with 1 infected individual? What do you think is going to happen? Try it. You tell me what you got. Anybody have something, an observation when they started with 1 infected individual?
Seems like there's really only 2 options.
It seems to draw the lines closer together.
So when I start with 1 infected individual, the time at which people get infected varies a lot. So it looks like everybody, in the end, it always goes to infected case. But let's try going to 60 maybe. But okay, well here there was 1 case where it didn't infect, where it died out. So that was, I guess, what you were saying about there were two situations, one where nothing happened and one where most people got infected. So, and the time at which the infection happens and peaks is quite variable. You can't really make I less than 1 because Gillespie is about individual. So what you see is that you get these big fluctuations. If I make both S naught and I naught small, like start with 100 susceptibles and 1 individuals, here I see a couple of cases where either the infection died out, a fairly broad range of cases where it didn't die out. And this is again for R naught 2.5, 100 initial individuals. If I start with an initial population of 10, I get things all over the place. Really, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 are not a very good approximation to continuum. And so it's not surprising that I don't see or recapitulate the continuum result. In this case, you'll notice after 30 days, the infection has always died out. There are always 0 infected individuals for these simulations. The final number of infected individuals, however, is enormously variable. It could be 9, 8, 5, 4, 3, 2, 1, or 0. And so if I have this small populations with 1 infected individual, what happens is quite, it's highly variable and doesn't look very much at all like this ODE prediction. And so there's some interesting questions you can ask about, well, this was for R naught equals 2.5. Let's make R naught bigger, which would be equivalent to making N bigger. Let's make R naught 15. 7.5. Well, that just speeds things up. But notice all those intermediate cases have disappeared. You either have everybody infected or nobody infected. I'm going to make R naught big. If I make R naught small, in this case, there's no infection. This is going the opposite way. Here, my R naught is 0.75. For R naught 0.75, my ODE says there's no epidemic. And so a few people get infected and then the epidemic dies out. In my stochastic simulation, that happens sometimes, but there's some cases where almost everybody gets infected, even though R naught was small. Because if I cough on you, and your probability of being infected is 0.75, and then you cough on 5 people, probability of each one of them being infected is 0.75, then there's still some chance of a chain of transmission going on for a while. And so in this case, you actually wind up with more infection in the stochastic case than you would otherwise. Typically, if R naught is larger than 1, the number of infections is lower in the stochastic case than you'd predict. In this case, when R naught is less than 1, the number of infections is more than you'd predict. And so there are quite a few things that you can explore with this that are pretty interesting. Again, knowing that infection really is stochastic. And infection really is determined by individuals interacting with each other. And so originally I had a set of exercises that we could do. I think it's more interesting. We could save some of those for homework, perhaps. I think it's more interesting probably to work on something together on one of these papers that you guys found. There was again in this there was a online talk that I found by Jamie Lloyd Smith from Penn State, which had a nice result, which was he looked at, actually Jamie, I don't know whether that's male or female. It's a name that could be either one. They looked at, these days, they looked at the case where you start out with 1 infected individual and what was the probability that the epidemic died out. And so as a function of R naught. In this case, R naught was less than 1. The epidemic always died out. Let's make R naught bigger than 1, much bigger than 1. So let's make N 10 times bigger. In this case, in these lines up at the top, the epidemic died out. In these lines below, it didn't. I'd have to plot it a little bit better to figure out which ones had, how many of them were those. I can look pretty well over here though. 1, 2, 3, 4, 5, 6, 7, 8, 9. Looks like 2 of them died out and 8 of them didn't. And so I could run this many times and calculate what fraction of the time the epidemic died out. And they in this online talk, they had a nice plot. If the probability, if R naught is less than 1, the epidemic dies out both in the ODE and in the stochastic model. If R naught is bigger than 1, the epidemic never dies out in the ODE model, but it has a finite probability of dying out in the stochastic model that decreases as R naught gets bigger. And so one thing you could do would be to run this simulation many times, say for S naught equals 100, I naught equals 1, change your R naught value and ask how often does the epidemic die out, defined by staying up at the top here, the total fraction of people infected less than 5%, for example, and then plot that as a function of R naught. And so that's something we could think about doing as a little homework assignment. I think it could be an interesting thing to play with. We usually think about the disease dying out as a good thing. Of course, if you're thinking about it from the point of view of the virus, the disease dying out is not such a good thing. Clearly, at a global level, if the virus is still there, it's not dying out. So even if it's dying out in a group population, it has to have spread to another population where it can proliferate. And so I think that's a homework problem that could be sort of fun to do. I wanted to talk a little bit about other model structures. One of them is a SIS model, which we hope COVID isn't. This is a disease that has where you have no immunity after you've had it. There are plenty of bacterial infections that are like that, and some eukaryotic infections that are like that. Common cold is an example of that. Coronavirus colds are usually don't give you any immunity or very limited immunity. Maybe it's worth having it not as a sort of a micro assignment, having people look up some examples of diseases which don't give you immunity. Unfortunately, a lot of sexually transmitted diseases don't. They're not viral, but things like gonorrhea don't excite long-term immunity. There are plenty of others that don't as well.
James, would you say if you have to take antibiotics, as in the case of STDs like those ones, that you do not develop immunity and then you can use an SIS model?
I don't know whether the use of antibiotics is necessarily the issue. So the issue is really how your immune system responds. Do you make antibodies to the disease? Do you make T cells to the disease? And are they effective in preventing the disease? And so I don't know whether it's the mode of treatment that's the issue. Certainly diseases that you wouldn't clear naturally.
Right. The reason you take antibiotics is because you don't clear them.
Right. So syphilis or gonorrhea were diseases that historically you live with for your whole life. Of course, HIV, even with treatment, is still a disease that you don't clear.
Right.
There's a viral disease that you don't clear. Herpes is a viral disease you don't clear. But those are more in the SI category.
Right.
Here we're talking about disease that you do clear, but doesn't leave you with long-lasting immunity. And so I guess that exactly how you characterize the disease is and I got distracted to look up some examples of SIS diseases. There certainly are enough. And they're certainly diseases that people get every year. I mean, influenza is a little bit different because you have some immunity to the strain you already were exposed to, but because of the variation of the viral coat proteins, you tend to not be immune to the new strains. But if people feel like adding that to their stack of things to do and make some suggestions, I'd be grateful for some ideas about SIS model. An SIS model looks like an SIR model, except the output is S rather than R. And the reason I wanted to bring these up was that this is a case where you really do have a non-trivial fixed point. Everything stays the same as before. R naught is beta over mu, and so on. But in this case, there are two fixed points. There's a trivial fixed point where S star is the total population and I star is 0, as you have no infected individuals to begin with. But I can also set ds by dt equal to 0. I set I naught to 0. That means beta over S naught times S plus mu is 0. In other words, if I star is equal to 1 minus mu over beta, that is 1 minus 1 over R naught times S naught, then I have a stable situation. And so my herd immunity criterion for SIR models is a stable, what's called an endemic. In this situation, I will always have 1 minus 1 over R naught of the population infected. So if R naught is 2, half the population will always be infected. If R naught is 20, 95% of the population will always be infected. And that's called an endemic. So you have a stable, steady state for the we haven't talked about stability, but it is a stable, steady state. A stable, steady state for the infection. If R naught is less than 1, then the disease dies out just like an SIR. Okay. I wanted to talk a little bit, because we're going to need them for the exercises that we're going to do now. We're not going to do these exercises, but they're written as exercises. For a little bit of computing, there are two very simple concepts that we're going to use to have things change in time inside of antimony. Our antimony specifications are, remember, not program specifications, they're model structure specifications, which is a little different. And so far, our descriptions of our viral replication models and our epidemic models don't have a concept of a loop or an if in them. And so we can't have something change on a condition. And so within our antimony syntax, there's a concept of an event, which is an if statement. And it has the form of the name, it can be named, so you can have it, give it name event 1, colon. And instead of using the word if, because that sounds too programmy, they use the word at. And the main concept was that it's a time event, but it doesn't have to be a time event. So the syntax is at, open parenthesis, logical condition, colon, what happens. And so, for example, suppose that I have A represent the concentration of a drug, and at time 15, I give 15 units of the drug. I would write at time greater than 15, A equals A plus 15. There's no plus equals in antimony the way there is in Python. So you have to write A equals A plus. Okay, does everybody follow that? The syntax isn't terribly complicated. You just have to remember that instead of an if statement, you use an at, and you have a colon to separate what the condition is from what happens afterwards. So that colon actually looks Python-y, because if you had a Python if statement, it would have a colon before what you do. Okay. So the syntax is basically event name, colon, at logical expression, colon, and then what you want to have as an output. And you can have multiple things happen before the end of the condition. Notice that it's not quite the same as an if statement because there's no concept of looping, or there is a different concept of looping in antimony from what you have in procedural language. If you write the condition at time greater than 15, a equals a plus 15, you might expect that, well, at time 15, this is true, and so you add 15. At time 16, this is true, so you add 15 again. At time 17, this is true, so you add 15 again. But then how often would you do it? I mean, time is a continuous variable, so at 15.1, it's true. So if this condition were triggered all the time that time is greater than 15, then at time 15, suddenly a would go to infinity because you'd be adding 15, 15, 15, 15 continuously. And so the default concept is that when the condition goes from false to true, the thing on the right-hand side of the condition is done once. So if I ran this at time 15, a equals a plus 15, a here is decaying. At time 15, I add 15 units and then it starts decaying. So that event is triggered once. Okay. If the event trigger becomes false and then becomes true event again, the event triggers again. And there probably are more elegant ways to do periodic events, but I tend to use something like this: at sine function of time greater than, say, a half, b equals b plus a half. That means every time time is modulo 2 pi, b will trigger. And so if I run that, I'll see glitch, 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 because every time that argument goes from false to true, it triggers. So it lets me do something that's periodic in time. I don't have to have an argument on time. I could have an argument on the value. I could say if a is greater than 50, a equals a plus a half. If a is less than 50, a equals minus a. And so I could actually have do something to a that actually changes the logical state of my function. And there's some slides which I'm not going to go over because I want to move on in the class, but there's some slides that are in the deck that I'm not going to show now that talk about how to do that. We also found a bug in antimony that Herbert and Andy are supposed to fix, which is that if you have two events happen simultaneously and they both reference the same variable, so I say at time greater than 15, a equals a plus 15. At time greater than 15, a equals a plus 10. You should wind up with a equals a plus 25 because both events should happen. Instead, one of the two events will happen and it's random. I don't think you're going to run into that today, but you should be aware that that's a bug. The specification for the language says that if two events happen at the same time, they both happen. And the reality is that with the code we have, when two events happen at the same time and affect the same variable, they affect different variables, you're fine, but if they affect the same variable, one of them happens and you don't know which one. Okay. If you want to read more about events, the documentation is there. And you can do things like delays with events. So you don't have delay equations. You can't say dx by dt equals something at a previous time, but you can say 10 seconds after an event occurs, do something. So you can have delayed events. The other thing that we should be aware of are time-dependent functions. If I write a equals 10, the value of a is set when I do my TE load a. If I say a equals 10, b equals 10 times a, the value of b is set when the parsing is done at the TE load a. So if I want something to change in time, and I don't want to write it in the form of a reaction, I can write a temporally variable function with the syntax colon equals. So that looks like C. So if I write K1 colon equals 0.2 plus sine of time, then the value of K1 will be continually updated to 0.2 plus sine of t. If I wrote K equals 0.2 plus sine of time, K1 would simply have the value 0.2 because it would be evaluated once at the beginning of the simulation when time was 0. Sine of 0 is 0. And so K1 would be set to 0.2 and left there. And so that's something that we'll be using, we would definitely be using today. In fact, something that looks almost identical to this will be something we'll use. And so I could now say a rate constant could be a sinusoidal function of time. Here I had a simple chemical reaction, a decay reaction, and I made one of the rate constants now sinusoidally varying in time. And you can see the sinusoidal oscillations of the chemical concentrations, and we'll do that. Okay. Things are a little bit more complicated if I have dependencies on variables. And I actually had the slide backwards. So Josh checked it out today and said I had it wrong, and we fixed it. So if I wrote something like K2 equals 3 times K1, and I want that to be implemented continuously in time, if I wrote in my antimony K2 equals 3 times K1, K2 would be given the value of 3 times K1 at the beginning of the simulation. And if I change K1, it won't update K2. If I write K2 colon equals 3 times K1, then K2 will update anytime K1 changes. And so then if I go in and I change the value of K1, say with Python, K2 will get updated. There are some complications of this, which are actually fairly confusing. I'm not allowed to go in and write m.K2 equals something in Python when I've defined K2 in this way. But I am allowed to go in and change K1, which changes K2. And this is not, and these functional evaluations are not done at the beginning of the simulation. So if I change an initial condition, it may or may not work. Those subtleties I hope we won't run into today. But if you're using this functional notation and you get a result that seems strange, print out the values and make sure that it's actually updating the way you want it to, because it may or may not. The last thing that I wanted to talk about was explicit ODEs. I've mentioned before that if I write arrow nothing, arrow goes to B, semicolon K1 times A, that's equivalent to writing dB by dt equals K1 times A. I can write explicit syntax K2 prime equals 3 times K1. That would be equivalent to writing arrow goes to K2 semicolon 3 times K1. And so if I want to write explicit ODs, I can do it. they do function a little bit differently in terms of initial condition specification and and updating. And if I write K2 prime equals 3 times K1, I have to give K2 an initial value. If I don't specify an initial value of K2, it'll crash. Because to specify an ordinary differential equation, I have to specify initial value. In the case of a chemical species, if I don't give it an initial value, it assumes it's 0. For ODEs, it won't assume it's 0. So it'll crash. And so that's something that we probably won't need today, but possibly will. So I wanted you to see that syntax as well. Okay. I wanted to just very quickly talk about some other model structures. You've seen in some of the papers we've looked at SEIR models, susceptible, exposed, infected, resistant. This means that there's a latency during which you're not infectious and you're not sick. SEIR models give you a slight delay before things happen. And they fit, in the case of COVID, they fit the exhibiting data a little bit better than what we've seen before. It doesn't change the basic SIR pattern very much. And we'll skip because we're going to jump directly into some models that do this kind of thing. I'm not going to go into the details now, but one can explore what happens. Basically, if the time you spend in E is long compared to the time you spend in I, then the results look a little bit different. You can also imagine that when you're sick in different phases. This is also very much true of COVID. The key issue with COVID is that you're infectious but not sick for a couple of days. And that's the time you tend to spread the disease. Once you get really sick, you're in bed at home and you're not spreading the disease. And so in this case, you have a Q phase or a quarantine phase when you're sick but not causing people to get sick so much. And so in this case, you'd have SEIQR. The I phase, when you're feeling okay but infectious, would be fairly short. The Q people might still be a little bit contagious, but not as much because they're not going around spreading the disease as much. And so then again, you can analyze the model structure. Not that much is different, but for given values of R naught, you'll get different results. And for different timings, you'll get different results. The last thing, and this is something we're going to do explicitly in a few minutes in one of the examples, is that if you have births and deaths, things change quite a bit. And so the classic model here is, of course, this only applies if you're operating on a fairly long time scale. So if COVID comes in and we develop a vaccine and we don't have any more epidemics, then it's not a big problem. But especially for what we're called childhood diseases, every year as children are born, you have people who are not immune coming into your population. So your S naught fraction is going up. I mean, your S fraction is going up, your susceptible fraction is going up. And if people are immune after they've had the disease and are dying off, your R fraction is going down. And so suppose you have an epidemic, sweep the population in an SIR model. Everybody's now, or 90% of the population is now immune. And so the disease doesn't spread. As time goes on, new people are born, they're susceptible. The disease doesn't spread, so they don't get the disease. And then after 25 years, for example, a quarter of the population is now susceptible. And now your susceptible fraction goes above 1 over R naught, and you have an outbreak. And so this is classic occurrence of epidemics, where an epidemic would sweep the population. Plague would be an example, although that may not be why plague behaved the way it did. Sweep the population, kill 30% of the population. and then disappear for 30 years. Because the people who survived had enough immunity that you didn't have epidemics anymore of that disease. When new people were born, they came in, the fraction of people who could get the disease would go up. Once it went up beyond the threshold, you'd get another sweep of the population. And so you'd get diseases that didn't have annual cycles, but they came back every 15 years, every 20 years, every 30 years, depending on what the thresholds were. So in these kinds of models, we take our basic SIR model, and now we add an input, which is births. And in this case, we're assuming that susceptible, infected, resistant people all give birth at the same rate, which is probably not right. And that would, in this case, if we had this and nothing else, our population would go to infinity because there are no deaths to take people out. We have a constant, we have a birth rate that's actually exponential. And so here we'll assume that people die. So we have a death rate, which is proportional to the population in each column. In this case, we're assuming that the disease, whatever it is, doesn't kill people. And so the death rate in our infecteds is the same as the death rate of everybody else. If we actually had people dying of the disease, then we would have a different D in this column here. Okay. We can write our equations in a very straightforward way. We have an extra decay term minus D inside of our I minus DR in our R term minus DS here. That's actually missing. So there's a typo in this equation. Minus DS here should be. And there's the birth term B times S plus I times R. Birth rate proportional to total population. Since we're a little bit late, I'm not going to have us do this exercise. We'll do that together in a minute. But if I run this one now, for a birth rate, which is very high compared to reality, which is 0.01 per day. That might be true for, let's see, that's 1 every 100 days. So if you're a cat, maybe that's a good birth rate, but it's not true for humans. Chicken maybe can do that. What you see happen is that if I start with a largely uninfected population, I start out with my epidemic going through as it would. And then as new people come in, my susceptible rate comes up again. And then I have a new smaller epidemic, and eventually things level out like an SIS model. And so in these susceptible, infected, recovered models with births and deaths, what I'll get is an endemic where I'll wind up with always having a certain fraction of the population infected, some fraction resistant, and some fraction susceptible. That fraction will be that same 1 over R naught that we saw before. But when I'm starting out, I'll get these oscillations, these damped oscillations. And those damped oscillations are very typical of these kinds of SIRS models with births and deaths. And again, I was going to do this example in detail, but one of the papers that people suggested has it, so I thought it would be more fun to do that. So that was what I had planned for my lecture part of this class. I would suggest we take a 5-minute break now. Did everybody, how many people, did people look at the 3, we sent out that list of the 3 papers. Did people have a chance to look at those papers at all? We had a sign-up sheet and nobody signed up. So I don't know whether that meant that you didn't have time to look, whether the Google Doc was uneditable, was there, what happened?
Some people sign up already.
Okay, great, good. So, and how does this, so why don't we take
no, but before we leave, so I guess since there's just 6 people, we'll
yes,
do 2 papers instead of 3.
That's fine, especially, I mean, did people sign up for all three or was it
No,
just?
just for two of them.
Okay, well, that's fine. We don't have,
That we haven't done, but that's fine.
that's fine. There's no problem with that. So what I'd like to do after the, let's take a 5-minute break. Josh and I will do a minute or two to organize things. And then we'll assign people to groups. And we'll put you to work trying to replicate the paper that you chose in the way we've talked about. Josh will circulate. I will try to circulate, although it's not always possible for me to circulate if I'm the host of the meeting. So I may be stuck outside here, in which case, if you want to ask a question, you may have to come out of the group to come talk to me. The other way to do it would only be to put one of the groups into the discussion, into a breakout room. So let me and Josh talk about that. Why don't we take our 5-minute break, and when we come back, we will assign people to papers. We'll work on these things together. And then we're a little bit behind. I'd hoped we'd get done with my lecture part in an hour. We'll have an hour and 15 minutes to work on the replicating the paper. And then at the last 15 minutes, you get to present what you were able to do to the other group. How's that? Okay. Okay, so I'll see everybody back in 5 minutes, 6:30. Josh, how do you think you want to work this?
Yeah, definitely assigning just one group to the breakout room so I can go there, work with them,
Right,
and then you stay with the other group here.
Okay. So which two papers did people want to do?
They want to do the simple COVID-19 paper. This is a link to it.
So maybe I should unshare now and you should share.
That's fine. So there's still, yeah. So this group is full already. So I guess all the other students will work on this one.
Okay, sorry, I can't see your screen is a little small. So let's see. So just tell me which one is the one?
So they want to work on the simple COVID model, this one.
Okay, fine. So we'll put who wants to work on the simple COVID model?
Jeffrey, Nico, and Tyler.
Okay.
And then Brian, Callie, and Mercedes are going to work on the Shalom Unity.
Okay. And let me actually make that. So now that I put them in there, I can't make them co-hosts. I was going to make them co-hosts so they could move in and out, but once they're in, I can't make them co-hosts.
They can move out. What they cannot do is
Move themselves back.
right, put themselves back. Right. So I'll go to the breakout room then. James, when I'm in, can I chat with you from the breakout room or not?
I am not sure about that. You can tell me.
Let me go.
No. Nope. No, you can't.
No, I cannot. But if you send out like a message, I can
I
chat.
can broadcast, yeah.
Okay, I'll be in that room.
Okay, so the one that we're doing here is, let's see, which one was it was,
Here's a link to the document again. And then here's a link to the Google Drive. You can find the papers there.
so the one everybody picked was the one I didn't do. Okay, are people back? Mercedes, Brian, and Collie, are you back? Collie's here. Brian, Mercedes. Probably better to unmute so we can talk. So my goal, and maybe it would be better actually, instead of my screen sharing, if one of you screen shared. And I'm certainly here to help, but I'd like you to try to work on this as much as possible on your own, with help. And I would suggest that maybe we could do is, somebody could maybe be a sort of a scribe, or people could work on it together. We could go through the paper. We could create a markdown cell to begin with, for example, a markdown cell where we describe what the model is about, and then start writing code. But to begin with, do the same kind of, spend a few minutes doing the same kind of analysis that we did for the other papers, to say what it's about, what it's trying to do. And I have to say that I worked up 2 of the 3 papers, and this was the one I didn't do. So I'm coming to this. I did read it a little bit and look at the equations and the results, but I did not actually do on my own this replication. So So you have to tell me what the best way to do this is. I don't know if, as I say, I've got the screen share here, but I don't have to be the one doing the screen share. I think it might be better if somebody else did it. And I advise.
I could share my screen if no one
Okay.
else wants to.
Great. Let me unshare then.
So should I just like have a blank notebook open to kind of work through it?
Yeah, I think it might be good to, if you could set it up a little bit the way I did it, where you have the screen, the paper on one side and the notebook on the other. That way, if people have single screens, everybody can see together. Assuming that you like that way of organizing things. I mean, if you've been secretly hating the way I had the screens organized for all of these things the past couple of weeks, please let me know. That split screen was a sort of a compromise. Otherwise, the things are smaller, but then otherwise you wind up going back and forth. I think it's nice to be able to see the slide or the image and the.
All right, so I'll share. Okay,
Okay.
so can everyone see everything?
That's
Yes.
great.
I can see it.
So this paper has it's about using shield immunity to reduce the spread of COVID. And that's when you take people who are recovered from COVID and therefore assumed to be immune, and you have them go out in public and do all the necessary interactions, like essential interactions with people. And the idea is that in having more interactions with these shields, there are fewer interactions with infected people, and that will stop the spread of COVID. So basically, it's the model that the paper uses. It employs 3 different variations of an SIR model, actually. So if you look here, there's an SIR model here, and it's pretty much what we've been doing before, except there's this added term called alpha. And correct me if I'm wrong, but I believe that alpha is the ratio of contacts that a shield person would have versus a non-shield, versus a susceptible person. So alpha equals 10 means that for every 10 contacts that a shield person would have, then a susceptible person would only have 1. So they did some experiments where they, or simulations where they adjusted alpha to see how that would impact the fraction of infected people, and those results are here. So as you would expect, as alpha increased, the fraction of infected people decreased. And then they had other versions of the model, which you have to scroll down to the methods section to see. There was this model called flexible shielding, which had an even stronger effect. They just changed the terms a little bit here. And then there was, what was it called? Fixed shielding, which is this model here, and it's just a little more complicated. So each model gets progressively more complicated, and I could look at so the flexible shielding, looks like it doesn't provide in a flexible shielding mechanism, recovered individuals have 1 plus alpha greater contact rates than susceptible or infected individuals. And then in the fixed version, recovered individuals have 1 plus a greater contact relative to their original baseline. So instead of, I guess, comparing to the number of contacts that susceptible or infected people would have, this is, I guess, just compared to how many contacts that shield person would have had normally, if I'm not mistaken. So I was thinking to try to replicate this paper, we could start with the simplest model, like the basic model, and then maybe try to do the others if we have time. And then we can look through the paper to see what values they used for like beta, S, and I, and gamma.
I think that's a great introduction. I'd say Mercedes and Brian, I'd encourage you to open up Tellurium and do this in parallel or semi-independently. So I think Kali's got a very good introduction, but why doesn't everybody try to have their own version of it? And you know, that way, if she gets stuck, you can help her out. If you get stuck, she can help you out. And
Okay.
I'll that way. Otherwise, because watching somebody code is not always exciting. On the other hand, it's great. I think that's great. Thank you for the introduction, Kali. That was great. It's a nice paper. And it's impressive that you can get published in the Nature journal with that SIR model. You had to be fast, though. You had to do it right away in March to do that. So, okay. Okay, great. Well, this is good. So I'll let you, Kali, you're doing a great job. So why don't I let you lead off, and maybe the thing to do would be to just to begin with, you could at least in the markdown make a markdown cell and type the equations there, and then we can and then go from there and see what we need. But I think in an hour we should be able to walk through most of. We won't be able to generate all of the. We won't be able to regenerate all those parameter plots and phase plots, but you could do quite a bit. I think in an hour, at least the first couple of models. And this would actually be a really nice example of something you'd put up on NanoHub, because it walks through the successively more elaborate models.
Yeah.
And the concept is really interesting.
Yeah, when I saw this paper, I'm like, this is interesting, but if there is evidence that we can become reinfected with COVID, I don't know how practical using the shielding method would be. So that was just something I kind of had in the back of my head while reading this.
Yeah, well, that's something you could do. I mean, we could do one of the simpler ones, and then say, well, what happens to that if you have R goes back to S at some rate? How does it break? That's what's called an SIS model. That you say, okay, you stay immune for 3 months, 6 months, a year. Does it break the model or not? That's actually a really interesting thing to do, and that would be a lot of fun. So you could do the SIR model for a value of alpha, which works. Then put in R goes back to S. Change the rate at which you go back to S and see what it breaks.
Right.
That would be fun.
So
Okay.
Is that the primary difference between SIRS and SIS? In an SIS model, you never really truly recover. Okay.
So you're never resistant.
Okay.
So that R word really means resistant more than recovered.
Got it.
So you're never immune. So SIS model, you go immediately from being infected back to being infectable. Whereas in an SIRS model, you have some time when you are at least partially immune. So that would be more like influenza, where you have some period of time where it's very unlikely you're going to get influenza again 2 weeks after you just had it, but 6 months after you just had it, it's not so unlikely.
Right. Okay. So I guess we'll all start working on this and chime in if we have questions and go over it.
Sorry, I muted myself. You guys talk together and decide the best way you want to do it. Mercedes, do you have an opinion about how you want to handle this?
I'm currently looking over the paper and I have a Tellurium window set up, so
Great.
I'm kind of down with whatever works.
Okay. We did in the summer. We did these a hackathon, a 3-day hackathon, where we tried to run it this way. It worked pretty well, but then we had 24 hours of interaction time spread over several days. So try to squeeze it into an hour is maybe, but let's give it a shot.
Sound good to me.
So I've kind of blown up the model here, so like everyone can see it, I guess. And then I guess once my plan was just once I had the equations written down to then go look for the values.
Yeah, that makes sense. I guess that means defining new parameters, though.
Well, you could write the model structure. I would type if the people know tech syntax for things.
So,
It's not the most convenient thing in the world. Beta, but some of the Microsoft stuff is the same. So \beta is the Greek letter, for example, and you use a $sign to define an equation. We can always go back and clean that up later. I would just type the equations here in your text box so that you have them available, so that when you're thumbing through the paper, you don't have to keep looking for the equations in your.
Or also, Dr. Glazier, I did have a question. I put it in the chat and then Jeffrey answered it, I believe. But is it possible to reasonable to maybe use a linear algebra technique to find the fixed points in a model? Like maybe for a more complicated model, find a matrix that represents your system and then find the kernel.
Bless you.
Well,
Sorry,
so
excuse me. Thank you.
that's good. So there is in fact something called find steady state solution in antimony. So there is get steady state should find the fixed points. But I tried it a week or two ago and on a model, and it didn't work. And so the answer is that in principle, yes, the antimony and tellurium have a steady state solver built in, but it can be a little bit finicky. If you really want to do detailed analysis of bifurcations and fixed points, I wind up going back to XPP aught, which means retyping everything, unfortunately, which is this package that was written 25, 30 years ago by Bart Ehrmantraut at Pitt, which is very, very old, but does that particular thing really, really well. But yes, if we go to the tellurium documentation, there is a steady state solver, which we could use. So I tried that out and I can confirm that it works. That if you set it up so that the epidemic sweeps and then you increase the value of alpha, as alpha gets bigger, you can turn off the epidemic. So it's nice to know that it does behave as advertised, but I'll let you guys try that.
All right.
One thing you may have to watch out for, they call the variables beta, gamma, and alpha. I would be careful of gamma. So instead of typing a variable named gamma, I would call it gamma 1 or something like that. Because antimony, if you run into a reserved word in antimony, it won't necessarily throw an error. It will just do the wrong calculation. I believe gamma is actually the gamma function as a reserved word.
Is beta like that too, or is it just gamma?
I think it's just gamma, but when I use a Greek letter, I tend to put a number after it to make sure that there's no, that it's not going to cause me a problem. So I'd recommend using beta 1, alpha 1, and gamma 1 instead of beta, alpha, and gamma, and then you're safe. I think they actually have a mistake, but you'll see. I think their definition of beta is wrong. But you'll run it and you'll find out.
Remind me, if I set the variable s equal to m.simulate, what does s contain?
So
Hmm.
so what are you trying to plot here? Oh, I see, you're trying to do the, I don't think m.I is the right, how is it? It's a dictionary entry. So I think it would be S, this square, S curly bracket. I think it's S curly bracket, square bracket, no, not curly bracket, S regular bracket. And then the dictionary key. So it'd be quote I. Let me check that. That one, Josh uses that all the time, and I tend not to use it, and it's better to do it that way. So let me try that. Because the output of the simulate function is an array, and I think it's a keyed array. So let me just see if I can remember how to get the key, what the key specification is. That wasn't it.
Hmm. BLT plot.
Yeah, so S definitely. S is a keyed array, and I have to remember how you specify the argument of a keyed array. That. Let me ask Josh. I would suggest not worrying too much about the normalization. So dividing by m.s0 probably isn't going to be. And then you can just do your basic plot.
Okay, so
The other thing you could do is Was that it? Did that crash or did that work? S square bracket, quote, square bracket. Hey, Josh.
Hey, so you do it is S square brackets and then the name of the variable, like time, like in the chat. But you have to be able to do that, you have to specify it when you simulate. So in your simulation line, right, you have to specify it there.
So even though the column names are there, you can't use them.
Yeah,
unless
you cannot
did
use
it.
them that way, right? So we have a problem with that paper, the paper we're trying to replicate there, because it's really a sensitivity analysis paper. So because they don't have the real parameter values. So what they do is, okay, we have this range of parameters, and then we do it's 6 parameters. We do 100,000 simulations because of the combinatorials, and then we compare the results. So we cannot do that.
Well, but you can build the model structure, right?
Right. That's easy.
And you can run some of the parameters and see what
Right.
they do.
Okay. Yeah. That's okay. That's a good idea. That's what I was going to suggest at the boundaries, and then maybe in the middles. Okay.
And here, here it's a little bit the same. I don't think we can do all the bifurcation analysis they did, but we could at least do, for example, this figure 2, we ought to be able to replicate that first part of figure 2A, we ought to be able to replicate.
Right. That one's simple. Right. Okay. I'll go there.
Okay. So this, I think you've just run into the thing that I was thinking was the case, which is if you look at their value of beta, their beta is not divided by S0. It's not divided by the number of individuals. And so I think they actually made a mistake in this paper, because if alpha is not 0, then R gets to be big, and so this works. So if you try the value of alpha not 0, I think it'll give you something meaningful. But if alpha is exactly 0, it doesn't work.
So do you
So
think,
try alpha equal 0.1 or something like that.
Yeah.
See what
do you think that's why when they graphed it, they did the fraction infected because the beta didn't divide by the total population?
right, but
Hmm.
I think they were sloppy. I mean, I think they actually published the wrong thing. Because if you divide beta by S0, you get their result.
Does anyone know what the value is for R?
Well,
As
R
in,
would
like an
be,
initial condition?
so their value for R was should be 0.25 divided by gamma, so that would be 2.5. R naught. Oh, sorry, R being the number of recovereds. I think they're assuming
Yeah.
0 to begin with.
Okay. Just checking, because it's not. I'm not sure what is happening.
Okay, well, we could certainly
fairly certain that I got it all correct.
Yeah. So yeah. So I was getting. I was not getting. So when I ran alpha equals 0, I get what they get there. That fraction infected curve, I get exactly what they get, provided I divide beta by S naught. Here, what you're seeing is everything is dying out. And I'm surprised it's doing that for the alpha equal to 0 one. Let's just try doing it by, let me try doing it by hand because I have to say, let me try it. So I had something slightly funny happening as well.
No, why? 1, 2, 3, 4.
Yeah, so if I divide beta by S0 and set alpha to 0, I get precisely their baseline simulation. So that works. And then the problem is if you make alpha non-zero, R now is not normalized either. So really R should be divided by S0 too. So in the denominator, it should be beta over S0. And then in that denominator here, 1 plus alpha times R, I guess the thing to do is divide alpha by S0 too. That would work. And if you divide alpha by S0, then I think it'll work.
Do they all need to be divided by S0?
Well, I don't know. No, gamma doesn't. Gamma, ironically enough, is not. And the only problem is when you try to sweep alpha in that loop, it won't be divided by 0. It won't be divided by S0 if you do it inside, if you do it in that assignment. So you have to do it inside the E1. So let me try S0 there. Okay, now let's try it. Yep, that's it. So so they were pretty sloppy. So if you let me get the annotate. So if you, yeah, that'll work there, and then you need to divide R naught by S0 too in here. So R divided by S0 at the end of the one.
Like right here in E1.
Yeah, yeah. And now it should reproduce that curve. At least it did for me. Let's see what's different. Beta times S times I. And you got divided by S0 times 1 plus alpha times R over S naught. 0, that's right. And then you set alpha 0, 2, 5, 10, 20.
Should I not
What?
reset all here?
No, that should be right. Let me, let's see, because I've got basically, I've got, let me screen share for a second and show you what I got. Did anybody else have any luck with this?
I am not having any luck with it. Okay.
Can you see my my tellurium now?
Yeah, we can see yours.
Yeah, that looks good.
So that's not, I didn't do exactly the same spacing they did. I did alpha between 0 and 18 and spacings of 2, but this looks right. The time scale looks right. The input, I didn't normalize by S0, although I could. So let's see, what did I define? I defined S goes to I, beta times S times I, which is what you did.
Oh. I had negative beta times S
Oh,
times I.
that, oh, so that'll, then you'll have, then you're creating more S's and fewer I's. Okay, try that.
I also had negative beta.
So why don't we go back to your share?
There we go. I mean, they don't look different though.
You want to reshare and then,
Yes.
let's
Alright,
see.
so I should probably do m.plot
Whoa.
like that.
And then you have to do a T dot show at the end.
That's kind of crazy.
There you go.
Okay.
Down. Good. So you found a bug in their paper already.
Well, you actually found it, but
Well, it's just to show you could be in nature and still have mistakes.
yep.
Good. Okay. So now what they did was they looked at the shield strength and the herd immunity threshold. And so how would that one work? That would be the epidemic size. So if you wanted to generate 2B, what you would have to do is find the maximum. You'd find the maximum academic sizes is actually R infinity. So why don't we, instead of plotting I, let's plot R. R's R at the end in your plot. Yeah, you could, or you could do it as a separate. Why don't you just copy? Yeah, there we go. Plot R instead of S. You don't have to copy that. That'll just it'll remember from cells. So plot R. Good, let's try that. Okay, so now you see that the final number of people infected depends on A. And so now, if you wanted to do that, and I had the code example for it before, what we do is you create a loop where you're going to, so if you're going to do that, we do the thing we did with parameter scans before, where you create an empty NumPy array, where you're going to put the value of alpha in the first column and the final result for R in the second column, and then you'll plot that.
Okay. Because we already have the alpha values.
Yeah, you'll probably want to have more of them, so you'll probably use a NumPy A range to do it at a finer scale. So let me do that in parallel with that.
Hmm.
All right. So, yeah. Yeah, that one's a tricky one to remember. So maybe, again, I can show you what I did. There are probably a lot of ways you can do it. You could create a list and do an append instead of having to do a VStack. I find VStacks are not the prettiest thing in the world. But I did result is NPy empty shape 02. I looped my alpha. I ran it. I VStacked my alpha. And then I plotted the result. And that was my result, which I think looks a lot like what they had. I can't see their result.
Yeah, they do have a downwards, like a slope like that, exponential decay looking slope.
Right.
Mhm.
Yes, in that one, I probably do want to normalize by
Very similar.
not.
Hmm. It's kind of interesting.
So. But that one, as I say, I had a slide to remind people of that code because the V stacks and H stacks are not supernatural to do. I have to look them up. Actually, this time I remember it, but I often have to look them up. But that one seems to be all right. So if people want to copy that code and try it out. Brian, were you able to get it to run looking at Colley's code?
Yeah,
Or
I was
say-
able to get it to run. I think I also just missed a typo again. But I was able to plot SINR, and I just wanted to see like how that would function. It was kind of interesting.
Oh, the R one is nice.
Mm-hmm.
Right. Mercedes, are you able to were you able to get any of this to work?
Yeah, I just didn't do the VStack thing. So I could only see like one graph at a time when I was trying to plot it.
I wish there were a less fussy way to do those parameter plots. I don't know of, I mean, again, you could do something like result equals nothing, empty list, and then you could do result dot append that.
Plot array.
I think that would work.
I ended up getting the result.
So I think that might be a little bit simpler. And then I think plot array will work. So let me try it. I can't promise. No. So no, so if you want to do it that way, then you have to use a NumPy. You have to use matplotlib to plot it. So that doesn't do what you want. So okay, forget that. So I'm afraid I don't have a simpler way than the VStack. So Kali, do you want to share screen share again and let's take a look? So we got one of the figures replicated, and we found a mistake. You want why don't you screen share again, Kali, and we can maybe look at the next figure we forget to see whether we can go from.
I can't.
Once you got one, sorry.
I'm saying I
Oh,
can't screen
I'm
share.
sorry. I'm sorry. First, I wasn't sharing my screen when I should have been, and now I was sharing it when I shouldn't. Okay, great. So that one worked. And it looks like it replicated their result just fine. And then let's see about one of their other models. So they have a slightly more elaborate model.
Yeah, we could go with the flexible shielding maybe.
Great. Okay, sure. Let's try that.
It looks like the only thing that changes now you're squaring this 1 plus alpha r term.
Okay, so that should be easy to do.
So if I just make that change, I run it, get a very similar looking graph, but it looks like now with this flexible shielding, the decrease is even more pronounced than up here, which is what they said. They said that the flexible and the fixed models had a more profound impact on it.
Yeah, I'm getting some more pronounced, like more flayed out kind of graph. If that makes any sense.
Yeah.
Yep.
Yeah, it cuts off faster when you do the alpha scan.
Yeah, and I can scroll back up to see if we can find what the results for the flexible model look like.
Yep.
It's kind of annoying to have them mention the model and then you have to go back to the methods to find it.
Yeah, well, that's probably nature has very strict limits on how long the main paper can be.
Okay.
And so they tend to force you into putting things into supplemental material. Okay.
Yeah, I don't think it actually says. I think it just says that there was more profound impact. Let's see.
Yeah, it's probably in their supplemental material, not in the main paper. That's why it's there you are. Yeah. So that's I bet that's it. So let's see, how could we do that one? We could do that one. The way to do that one would be if you in your code, if you created a second, what was the name of your second model? If you in your Jupyter notebook, if we go back up, when you created the model with the square, with the flex one,
Yes.
scroll up a little bit. So if you call that, say, flex shielding instead of basic shielding, and then call it flex, and then why you load it as M2 instead of M1 there. And then you have to change everything there. And M.alpha too. Yeah. And now you could actually do something a little tricky, which would be if you copy your 3 lines M2, M2S, or actually just, yeah, those 3, and paste them back in underneath. And get rid of the M2 and make it back to M for the first one. And then instead of calling it S, call it S2 and that where you're doing the M2. Okay. And now you could make it, instead of making your empty shape 0,2, make it 0,3. And then stack on M.R over M.S0. So that guy, M.alpha is fine, but copy the rest of that line and add it back in, getting rid of the 2's. So that you don't know. I just here. I mean, there are a lot of ways you can do it. I'm just free associating here, but just copy this here, put a comma, M.R over M.S0, because you've just calculated that here. And I think that will give you these 2 lines. Let's try it.
Okay. Now we have to rerun some of this.
Did it fail? No. There.
Yep. That looks like it worked. This is M2. This is just M.
Right. So we could, if we were being fancy, we could put labels in and things like that. But for the purposes of today, I think that's not bad. And I'm not sure what their fixed shield mechanism was, but
Here they do like 1 minus 1 plus alpha.
so we could basically just grab the whole thing again and try it. Add a 3rd one, and plot them all in the same graph. Then you've basically replicated the whole paper in an hour.
Yeah. And then they do some other stuff with like an age structured model and other
Yeah.
optimizations that they have in.
Yeah, that one would take a little more time, but for an hour, I think you did pretty well.
Oh god, okay, so we have beta times S times I times 1 minus 1 plus alpha times R divided
Eh,
by S
F0, yeah, it's always going to be our time. They
All of
messed
that
up.
is
They
similar.
were inconsistent. They didn't write that right.
Yes, I think that's right.
The denominator is different. It's just 1 minus R, no alpha. Yeah, there you go.
Is it? Oh wait. Okay.
That's right. Yeah, that's right.
Yeah.
That should be it. And then you'll have to change the names of everything too.
Yeah.
And I guess if you want to do it, you'll have to add one. You already added the 4. Yeah, so.
Mhm.
Flex shielding has to be changed to fixed shielding where you say M3.
Yeah, I was gonna say there was a typo.
And then you have to add that one extra one in your result stack. The good thing about this crappy VStack thing is once you've got it once, changing it's not so bad. Okay, let's give it a shot. I think that should work.
Right, let's see.
Yep,
Well, yeah.
good job.
Full paper in an hour. And we found a bug. Look at us.
It's pretty endearing.
Mercedes, was that okay for you? You've been sort of quiet. I hope it wasn't boring.
No, I'm just kind of quiet.
That's okay. You have the right to be quiet. I just, you know, I don't, it's hard to know sometimes if you're able, if you're following it and able to get through it. I think that's good. I think maybe Kali, you can download it. Maybe if you're willing to add some text to the markup cells,
Yeah.
then we can distribute that to everybody. Do you think, do people want to work more? Or are we ready to meet up with the other group and see how they did?
Should probably meet up with the other group. I mean, it's already 7:41.
Yeah, so
We only have about 14 minutes left.
yeah, so I better tell them that they should. Okay, breakout room. Okay, broadcast.
Get over here.
Oh, that makes me feel powerful. I can close them and pull them back in involuntarily. Boom. Hey, everybody, how'd it go?
Well, I would say it was a little bit messy because it's a lot of sensitivity analysis, really. So, like some of the methods they are using to perform the sensitivities, I'm not familiar with.
Okay, I guess Kali, do you want to speak for you and, well, you shouldn't have to speak for everybody, but Kali, Brian, and Mercedes, do you want to tell us what you got? And then these guys can tell us what they were able to do or not?
As in, like, how did we do on the attempting the plotting?
Yeah, just show us what we just went over it, but they haven't seen it, so.
Okay. Basically, we encountered we were trying to plot the
I
entire
guess,
thing.
are we screen shared? You have to screen share. Kali, I guess you'll have to screen share
Mhm.
to show the paper and the.
So this paper talks about an epidemic mitigation technique called shield immunity, where you assume that people who are recovered from the disease have immunity, and you can introduce them back into the community to have like public interactions, so that susceptible and infected individuals have more interactions with those recovered individuals who are assumed to be immune. So if an infected person interacts with an immune person, that infected person doesn't transmit the disease to the immune person, and then a susceptible person won't get the disease from the immune shield. So this paper considers 3 variations on the SIR model to show how different levels of shield immunity change the fraction of infected individuals. So here's the basic model. It's an SIR model, except they introduced this new variable called alpha, which is what they call the contact ratio, which is the ratio of the number of contacts that a shield recovered person would have compared to a susceptible or an infected person. So alpha of 10 means that for every 10 interactions that an immune shielder would have, a susceptible or infected individual would only have 1. So we actually found a bug in this model. So they don't divide their beta value by the total number of people in the population. So initially, when we tried to use this model to get these results, we ran into some difficulties because in the equation that they used, both beta and R, the number of recovered individuals, should have been divided by S naught, or like the total number of individuals in the population. So that's one adjustment we had to make.
Mhm.
But then we kept the other variables as is, and we swept for the same alpha values that they did, and then got a very similar looking graph.
Yeah.
Although we did total number of infected individuals, they did fraction of the population infected.
Great. And Brian, maybe do you want to explain the other graph we generated, which would be that second one? So, Kali did a great job introducing it. Now let's turn it over to you to explain the second one.
The second one,
The second graph, yeah, that would be the B picture. That
the B picture. Hold on.
Yeah.
This was basically the unaffected to epidemic size, right?
Mhm. I don't want to put you on the spot. If you'd rather Kali keep going, that's okay too. But you had started talking and then you didn't get to talk. So.
I guess I don't really understand it as well as I am supposed to, but I mean, given the graph, it seems like it basically is just a graph of unaffected to the epidemic size, and essentially, like, there's a place in the graph that we can check the herd immunity. Callies, there's probably something I'm really missing here.
So the x-axis is the alpha, is the shielding rate.
Right.
And so what it says is that if 0.9% of your population would have been affected without shielding, if your shield strength is 20, only 0.2% of your population get infected.
Oh, okay. Yeah, that makes much more sense.
We didn't label the axes. Okay, and then do you want to scroll down and show the final result where you had the different models and did the same thing? Kali, if you want to talk that through, or Mercedes, either way. Okay.
Mercedes, do you want to say something about this?
I mean, honestly, I think you were going pretty strong, so you can just keep on going.
Okay. So So that was the basic model, the one that we just showed you. There are also two other models that are variations on the basic SIR model we saw before, versus what they call the flexible shielding model. It's a little bit different. We're not going to get into the reasons why. And the flexible shielding model has a bit more of an impact on the trajectory of the total peak number of infected individuals. So these peaks are a bit lower than they were before. And for the relationship between alpha and the proportion of people who get infected, this orange line is the original model and this blue line is the flexible shielding model. So the flexible shielding has more of an impact than just the basic model alone. Finally, we have this fixed model, which is even more complicated. The peaks are dampened even more, and the fixed model has even a lower proportion of the population being infected as alpha increases. So here's the basic model, the flexible model, and the fixed model.
And I guess one thing to think about is in that picture that you've got there, the idea that your shields could be 20 times as sociable as ordinary people is sort of hard to believe, but the idea that they could be 3 times as sociable may be not so hard to believe. And so for the green line, if your alpha, which is sort of that sociability number, is 3, then you've reduced your infection by a factor of 5. So in principle, if you could somehow implement that fixed shield reality and get an alpha of 3, you would actually knock your infection down a lot. Whereas if you use the base model and had a value of 3, still 60% of your population would be infected.
Yeah.
I don't know how realistic either of those things is, but that's the takeaway is that fixed model would have protection even for a realistic value of alpha, whereas the base model wouldn't. But that's I guess the main result from this.
Yeah,
So I'm very. Go ahead.
The other thing to note here that the paper talked about is that you could use this shielding method in conjunction with other techniques like social distancing and like quarantine. But if you do use social distance and quarantine, then that decreases the alpha value because the person then has fewer interactions. So then I guess while you could still do the shield immunity, it wouldn't have as much of a noticeable impact.
That's a catch-22, huh?
Yeah.
That's interesting. Well, that's great. Well, I think so that's what we were able to do. I think I was very impressed that Mercedes, Brian, and Collie were able to get that far with that. So Josh, Tyler, Antonio, Jeffrey, even if you don't have a result, show us what you didn't get.
What about I let them explain the model? I'll share my screen. I'll let them explain what the model is.
Sure,
And I'll
sure.
show what the results are
Sure.
and what we did.
Sure.
I guess I can kind of give an overview of this model. So this is a simple model of the COVID-19 spread. So it's got a ton of these different phases. So basically your S is going to be your susceptible. Your L1 is your initial incubation phase where you are still non-infectious. And then they added that L2 node in there where you are still incubating the infection, but you are infectious, as data has shown. And then it kind of splits between asymptomatic and symptomatic infectious. We looked up some results to plug into the parameters, and it was about 45 to 50% of cases were asymptomatic. And so that's just one of the parameters, a fraction of asymptomatic individuals. And then they both progress and into the R, the recovered phase. So we implemented all those equations and their parameters, and that is the overall graph that we got. And then we performed basically a comparison to see if we scanned over the value of pi, which was the fraction of asymptomatic cases, and if we scanned over the value of beta, which was your transmission coefficient, how that would affect the peak number of infections in this cycle. And so the first graph shows the relationship between, that's the pi value and peak infections. So as you can see, as your yeah, wait, sorry, I'm still, we forgot to put labels on that as well.
Right. Yeah. But yeah, as the fraction goes up, of the fraction of asymptomatic goes up, this is this.
Right.
And sorry, the vertical axis is the total number infected?
The vertical axis is the maximum of the IMAX.
IMAX. Okay.
Right.
So it's basically flattens the curve.
Right.
It doesn't say what fraction total infected. Okay.
And then the second graph is the same kind of relationship, except we are scanning through beta, which is your transmission coefficient.
So
And-
that one looks like something funny happened.
Right. So the reason we chose those two is because they say in here that the model, were the two parameters that the IMAX was more sensible to.
So could you rerun that now with more intermediate values for the
Yeah.
second one?
So actually, also the reason I chose 7 is because they did a full combinatorial where they scanned from the. I mean, okay, I can change it, right? But the range, they did it over 7, and then the full combinatorial. That would imply that we would have to run 100,000 simulations. So we definitely cannot do that. But
Yeah, it's just because the curve is so sharply peaked around 0, you weren't really seeing the curve shape with just 7 points.
Right.
Everything interesting is happening on the left of that graph.
Right. But something that they looked at interestingly here is that they looked at the cumulative curve, at the cumulative cases curve. And as they simulated the model, they looked for different parameter values. They looked at the variability in that curve, and then they quantify which parameter was variability in which parameter was contributing more to variability in the cumulative cases as time goes on. And here what they show is that at the beginning, most of the incubation time has a lot of at the very beginning, the incubation time contributes a lot. Whereas later on, pi, which is a fraction of asymptomatics, contributes a lot, and beta.
I'm kind of wondering how much I believe the fact that as the proportion of asymptomatic individuals rises, the number of peak infections decreases, because I feel like a significant portion of infections are caused by asymptomatic individuals not knowing that they're actually infected. So that might just be kind of a flaw of this design, is that maybe the data doesn't show asymptomatic individuals having a higher infectivity rate because asymptomatic individuals aren't going to get tested. So
is the assumption that the asymptomatics have the same infectivity as the people?
No, they do not. They have half of the infectivity.
Half. So maybe that makes a difference. In other words, if the asymptomatics are highly infectious, like being half as infectious is still quite infectious, then they might dominate. Whereas if the asymptomatics were very low infectious, then I'm always, mainly because it's the thing that's been modeled the most, I'm always coming back to HIV infection. So with HIV infection, the probability of infection through one sexual contact is much lower if the viral load in the people are lower. And so in that case, the people who are asymptomatic because their virus is under control are much less infectious. They're still somewhat infectious, but they're a lot less infectious. And therefore, that number is probably really important to the way this plays out. So if the asymptomatics are as infectious as the asymptomatics, then probably the asymptomatics will dominate. If the asymptomatics are 0.1%, 1% as infectious as the symptomatics, then probably the people with symptoms dominate. So that would be very interesting. What you bring up is very interesting, and one could study how that assumption of what the ratio of infectivity is affects these results.
Right.
That would be a really interesting thing to study. Julianne and Sylvia, I don't know, we could maybe. I haven't talked to them in a long time. The last time I saw them was in Morocco, of all places.
And I do have to say that the reason they do this is because they don't know, they assume that they don't know any of the parameters.
Right.
So they basically try all possible combinations to see how each parameter is affecting the dynamics of the infection.
I mean, this question again, and I don't know how much we'll be able to do it in class. This question of how you deal with high dimensional models where you've got lots of parameters is there, just the way we were talking about optimization and the fact that there isn't any guaranteed way to find a global optimum, a global minimum for an optimizer. When you're dealing with very high dimensional parameter spaces, there's no free lunch. If in principle you have 100 zillion combinations, and you really want to know what's happening, sometimes you have to run 100 zillion simulations, which you can't do. And so there is this tendency to do these Latin square sort of sampling approaches, and those often work well, but they're not guaranteed to. You could be unlucky. In these relatively simple quasi-linear models, they tend to work pretty well. So I guess I'm sorry if it was frustrating. I hope everybody was able to get something out of this. It looks like you were able to replicate the basic simulation structure without a problem and get some interesting results. And so, if nothing else, I hope that people feel less intimidated when they read a paper. It's not so bad to sit down and in an hour you can at least replicate some of the basic results. You may not be able to get all of it, but you can go in and get some basic results. And as Mercedes, Kali, and Brian found out, sometimes the papers have bugs, like they wrote the wrong equations.
Mm-hmm.
And so it's important to develop a little bit of intuition about when the problem is on your end. I typed the wrong thing. And when the problem is in the paper. Just because it's published in a Nature journal doesn't mean it's right. Now, in their case, the way it was wrong was relatively minor. And I even think I know, because of how mathematical biologists work, how that mistake got propagated into the paper. But I hope that was a worthwhile result exercise for you. It was sort of fun.
I wanna say that we did replicated before class. We did replicated the third paper that is the list, RSV virus, and I just posted a link if in the chat if you guys wanna have a look at it and see how we replicated that one, because that one we did replicate it almost completely.
Ironically, all the things I talked about events and functions and this kind of thing were for replicating the RSV paper.
Right.
But I thank you for being willing to do this experiment. You could give us feedback on whether it was a worthwhile thing to try to do. If you say it was not worthwhile, we won't try it again. If you think it was worthwhile, maybe we can find one of these when we move now to. My plan was now that we've had some experience with the epidemiological modeling. I was planning on next week going and starting on modeling the virus inside of the body. going back to viral replication and immune response in the body, if that's okay with people. I mean, one could easily spend a whole semester on epidemiology, but I think as an introduction, it's nice to see some different problems. So my plan was that we switch to the viral replication models inside the immune response and viral replication models next week. Structurally, they don't look very different. Nothing will be dramatically strange. But they do have some additional behaviors that we haven't seen yet. So, all right, we're over time by 5 minutes. Any final comments, questions? You're free to speak now, or to send us your emails, comments, questions, suggestions. And I do thank you for being willing to do a little experiment of trying to do the replication in real time.
I was going to say that tellurium is becoming less foreign to me now.
I mean, the tellurium part of it is certainly not trivial, but for me, the blasted V stacks and H stacks are still the most annoying thing.
No, yeah, I agree.
And that's a Python annoyance. Okay, well, thank you, everybody. I'll see you next week. If people want to talk about projects, please email me. I'm eager to talk to people individually about projects and make sure that you can get them targeted. Again, some people are doing fine and have everything done, but I want to make sure everybody is on target with that. If the thing you uploaded for your project proposal was, in your view, done, that's fine. But I'm giving people who are not as finished with that the opportunity to have another week to work on it. But I really do want people to try to finalize their project ideas by next week, because we'll have to start working on that. And there was the little homework that was due today. We may have a simple exercise in the stochastic models based on what we did quickly today. Fill in a little bit of that. Maybe one SCIR model, although you've done it now in more detail, so I'm not sure that's worthwhile. And then I did send around a paper about the viral replication and immune response modeling. There's this nice paper that by Erica Mohan and Jason Shoemaker that has a nice review of some different models that we can play with. And frankly, any of those would be a good model to run. So we can sort of work from there. And I would encourage people, if you are not familiar with how the immune system works and how viruses replicate, to do a little Wikipediaing to familiarize yourself with that a little bit. Okay, thanks everybody. I appreciate it. And if there aren't any more questions, I'll see you either in one-on-one meetings or next week.